So today we get to talk about one of my favorite topics ever. But before that, here's the meme of the day, freshly made from last night. And there's a bonus because it's my last class. So I made 2 memes of the day. Is it good?
Yeah.
Nice. Perfect. So today we're going to be talking about mycorrhizal fungi. As you guys probably already know, that's what I do for a living. So I work with mycorrhizal fungi, good morning, Cameron, working on the side of carbon sequestration. So we're going to talk about the types of mycorrhizal fungi, all the details around this relationship, what benefits can the soils have, can the plants have from this fungus, and how can we manage our soils to get the best out of the fungi, okay? But before we move forward, do you guys know what is the difference between symbiosis and mutualism? Yes.
Is it mutualism, they both benefit, but symbiosis, they're like feeding off of each other?
Almost that. So mutualism, correct. So mutualism is a close relationship between organisms where both of them are benefiting, okay? But symbiosis only means that you have 2 organisms living close together. So it can be detrimental or that can be beneficial. So parasitism, for instance, is an example of symbiosis, not the good one, okay? Mutualism is the good example of symbiosis. So today we have a symbiosis, but there is also mutualistic. But before we move forwards, every time you hear the word myco means fungus, every time. And rhiza means roots. So mycorrhiza is a type of microorganisms that live inside of the root systems of around 90% of all the living plants. And they live in a symbiotic relationship where the plant provides the carbon that the fungus depend on. It's very important for you guys to remember that fungus cannot do photosynthesis, okay? So in mycorrhizal fungi, they cannot eat on their own. They need someone to feed them. What happens here is that when they get that relationship with the plants through the process of photosynthesis acquire that carbon and send it straight to the fungus. And the fungus, in order to help the plant, they can expand the root system of the plant up to 300 times to explore areas of the soils that the plant would not be able to do by itself. And it releases more nutrients to the plants, but also water. We're going to talk about the other benefits too. What is another thing that is extremely important when we talk about ectos? The morphology of the host plant root is changed. So we have several pictures here that show you that if you go to the field, you can go outside of Throckmorton right now. There is a pine tree over there. You can dig up the roots and you're going to be able to see those structures. So you can see them with the naked eye. And I did collect some of those samples for you guys, so you don't need to be digging stuff out in the cold. So these came from pine roots from outside Throckmorton and some from the Blueville nursery. So what I want you guys to notice is that when we have ectomycorrhizal fungi in the soil, you can see with the naked eye the branching structures. So I'm going to be passing this around, and I'm also going to pass, yes?
So the difference between like without the ectomycorrhizae and the like without it and with it is that it branches more?
Yes, it branches, but it branches in a specific way. I'm going to show you in the next slide, okay? But I'm going to be passing this around so you can see in person how it looks like, okay? These structures that they form that allow you to see when the fungus is present, it's called the menthol and the Hartig net. So as you can see here, the menthol is just that layer of fungus that is going to be outside of the root. The Hartig net is going to be the fungus that penetrate in between the root cells. And the word in between is really important. As you can see here, it's not coming inside of the root cell. It grows in between, okay? But when you go to the field and you dig them up, this is what you're going to see. So they inhibit the growth of root hairs, and they start forming branch-like structures in a similar way that we saw with rhizobium. Remember when we talked about nitrogen fixation? The root hair will curl, they will start reproducing and producing structures. That's what happens in here. And right next to the structures, you can see the fungal hyphae. We talked about hyphae briefly in the beginning of the course, but hyphae is going to be a thread-like structure that looks like a hair that is going to expand in the soil. That's why it can expand the root system up to 300 times. Yes?
How do you tell the difference between like a root hair and the hyphae?
That's a very good question. So, usually when we look at the microscope, we can see that the hyphae penetrates the root. Even in ectos, they don't penetrate inside of the cell, they do in between. And the root hair, it grows outside. So let me draw an example for you. So you have your root system right here. The root hair, it comes from the root. So when you look, you can see that it's part of the root. When you look at a hyphae, it cuts through the root. Does it make sense? So this is just a part of the root, and the hyphae got introduced to the root, okay? And hyphae tend to be way longer than the root hair too. Sounds good? Now let's talk about the benefits to the soils. And I brought 2 soils here to show you guys. And I'm not gonna tell you where did it come from. You're gonna tell me. So I got 2 soils right here, and I'm gonna add water to that. So let's simulate a rain.
We're gonna look for
Look at the color of the water. So one of these soils is full of mycorrhizal fungi, and the other one is depleted. See the difference? So here we have soils from the native prairie, collected from Konza. And you can see that we almost don't lose any soil particles. And here we have soils from conventional tillage.
That's crazy.
What is the difference?
The soil is aggregated.
Yes, so mycorrhizal fungi is the main driver for soil aggregation. When we have mycorrhizal fungi, they enmesh, they hug the soil particles together so tightly that we don't lose it when it rains. So when you till your soils, the chances of losing the topsoils and your nutrients are way higher than when compared to a soil that is full of mycorrhizal fungi. And if you want to see how soil aggregates look like, you can see here aggregates from a no-till system and from conventional tillage system after 34 years of tillage. I'm going to be passing this around. It's from the long-term experiment of Dr. Rice. So remember in the beginning of the class that I told you that the benefits to the soils can be even more significant than the benefits to the plant? And that's true. So as you can see in those pictures, the hyphae of the fungus is what keeps soil aggregates together. So if you deplete your population of mycorrhizae, you're also losing soil aggregation. Remember the dog, the chihuahua from the beginning, from the meme of the day? That's why they were so upset when you till the soil. Because when you till the soil, you're breaking down those structures. So you're exposing the aggregates to microbial degradation, okay? Neighboring plants that are connected by mycorrhizal, if the first plant gets beaten by an insect, it immediately texts the other plant through the hyphae and tell them, brace up, insects are coming. So the other plants start producing compounds that will kill that insect or repel that insect before it reaches it. It's pretty cool. The communication, the gossip in between plants when they have mycorrhizal, very effective.
What I'd give.
Yes.
So for the inoculation, you said earlier that it's hard to inoculate them because you need the plant.
Yes.
So how are you getting the spores?
Very good. So what big companies do is they use what we call a trap culture.
Okay.
So they grow sorghum to produce the spores, and then they kill the sorghum and use the roots of the sorghum and the soil around as a source of spores.
Okay.
And what do they do in big companies is that they isolate those spores and sell it.
Oh, okay.
And at the field, we apply it in furrow.
I see.
So we dilute it in water and apply it with the seed.
Okay, I see.
And it's not very expensive, actually. For horticulture, it's very cost effective. For crops, we're still studying to see if it's good or not.
So